Morning, everyone. I am Therese Ijala Moran. Today, we're going to talk about protein synthesis, transcription, and translation. Protein synthesis is the process in which cells make proteins. The purpose of this is to simply create a polypeptide, or a protein made up of chains of amino acids.
Instructions for making proteins with the correct sequence of amino acids are encoded in our DNA. Our DNA is found in the chromosome, and our chromosome remain remain in the or in our nucleus. But proteins are made in our in a ribosome in the ribosome in our cytoplasm or on our rough ER. So if the DNA is in our chromosome that stays in the nucleus, how do the instructions of DNA get to the cytoplasm? So this is because of the nucleic acid RNA, which can squeeze through the pores in the nuclear membrane. RNA carries the informations from DNA in the nucleus to a ribosome in the cytoplasm, and then helps assemble the protein. In short, DNA contains genetic informations which will be carried by our RNA carried by the RNA to our ribosome, carried by the RNA to a ribosome, and RNA also helps assemble the protein. So from DNA, carried it will be carried the genetic information will be carried by our RNA, and RNA will help assemble our protein. So we have two there are there are two phases for protein synthesis. The first one is transcription and the second one is translation. So transcription phase. Is the transfer of genetic instructions in DNA to mRNA. Transcription has three steps. The first step is the formation of hnRNA, second is the formation of mRNA, and the third step is mRNA enters the cytoplasm. So, first step is the formation of hnRNA. hnRNA or heterogeneous nuclear RNA is formed directly by DNA transcription. So what will happen in our step 1 or in our formation of hnRNA is that DNA in the nucleus partially unwinds to allow a strand of hnRNA to be made. DNA is a double helix, meaning it has two strands that are coiled together. And this is unwinded by the RNA polymerase during transcription process and exposes some bases or genes. Everyone, we need to remember that these bases or genes has its corresponding partner. In DNA, uh, our so these bases or genes are adenine, guanine, cytosine, and thymine. In in DNA, our adenine is usually paired with thymine. But if we are going to talk about RNA, like today, we will pair adenine with uracil, and thymine will be paired with adenine, and guanine will be paired with cytosine, and cytosine will be paired with guanine. So now that our DNA has been unwinded by our RNA polymerase, then we can say that this strand right here is our template strand, and the other strand is our informational strand. Our, our template strand is where the ribonucleosides align along, and it is also used for HN RNA synthesis. And our informational strand is not involved in the RNA synthesis. So, about 10 base pairs of DNA template are exposed at a time. Like this one right here, it has 10, 10 base pairs that has been exposed after the DNA has been unwinded by the RNA polymerase. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So, so free ribonucleotides align along one of the template strand, forming a new base pair. So as I have said earlier, these bases has its corresponding partner. So we should pair them correctly. Like this one right here. This one, the colored ones, is our HN or our newly formed HN RNA strand. Guanine or our cytosine rather is paired with guanine. Adenine is paired with uracil, and so on and so forth. Not only does the RNA polymerase unwinds the DNA, it will also it is also involved in the linkage of ribonucleotides to the growing hnRNA molecule. Okay. This process will continue until RNA polymerase encounters a sequence of base that is read as a stop signal. This process will continue and continue so on and so forth until the mRNA will become long, but it will only stop if our RNA polymerase encounters the stop codons UGA, UAG, and UAA. So once the RNA encounters a stop codon, the newly formed RNA molecule and RNA polymerase are released and our DNA reform to its original double helix. So we have now a newly formed hnRNA. So we have finished the first step in our transcription. So let us now move on to our second step, which is the formation of mRNA. mRNA is also known as our messenger RNA. It carries instructions for protein synthesis. In this process, introns are removed from the hnRNA strand because not all bases in a gene convey genetic informations. Also, a gene is segmented, meaning parts of it contains exons that which conveys genetic informations and introns which do not convey genetic informations. Introns are not needed for protein synthesis as they have no genetic information and only interrupt the genetic message. So, so introns are removed from the hnRNA strand through the through the process of splicing. Splicing is the process of removing introns from an hnRNA molecule and join remaining exons to form an mRNA. This is facilitated by the snRNA, which is our small nuclear RNA. hnRNA contains exons and introns. Introns do not convey genetic informations, so it is not needed in our protein synthesis because it will only interrupt the genetic message. So, through the process of splicing, which is facilitated by our snRNA, splicing will remove the introns and then join the remaining exons together, forming our mRNA. It's like that our hnRNA has been edited, edited because the or because the errors has been removed. So we have now finished our our second step, which is the formation of mRNA. So moving on to the third step, which is mRNA, which is moving on to this third step, mRNA enters the cytoplasm. The mRNA leaves the nucleus and enters the cytoplasm. Our mRNA can leave the nucleus because of the nucleic acid RNA, which can easily pass through the nuclear membrane. So, I believe we have finished our transcription phase. So, let us now move on to our translation phase. Our translation phase, what will happen in our translation phase is that mRNA are deciphered and a particular protein molecule is synthesized. The site for translation phase is in our ribosomes. Translation phase have five steps. The first step, the first step is the activation of tRNA. Second step is initiation. Third is elongation. And fourth and fifth is the termination and post-translation process. So, this is what our this is what our tRNA looks like. The tRNA or transfer RNA delivers amino acids to the site for protein synthesis. This one right here, this one right here is our tree end. Our tree end is where amino is the tree end is our amino acid attachment site. And this one right here that is colored with the purple is our anticodon. Anticodon is a three-nucleotide sequence on a tRNA molecule that is complementary to a codon on an mRNA molecule, meaning it is paired to a specific codon that matches its nucleotide sequence. As I have said, right, that the bases has its own pair. And our anticodon contains three bases. And these bases will match our codon in our mRNA. So if if the anticodon do not match or do not pair with the if the base and the anticodon do not pair with the codons in our mRNA, then it cannot attach to the mRNA. So let us now proceed to our step one, which is the activation of tRNA. It this this step has two process. First step is the first step is an amino acid interacts with ATP to become highly energized. For example, the amino acid MET. These are wait. No! Oh my god, nawa. Today. So our tRNA is not activated if there is no amino acid attached to it. So the activation of tRNA composed of two process. First process is that an amino acid interacts with ATP to become highly energized. For example, the amino acid MET, it will interact with an ATP to become highly energized. Second step is that highly energized MET will form a covalent bond with the three end or the attachment side of the amino acid of a tRNA molecule. So our for example, that is our MET, it will attach to the three end of our tRNA, and once covalent bond has been formed, then our tRNA will become activated. Also, amino acid tRNA pairing is governed by the tRNA ligase. The amino acid that the tRNA carry are determined by codons rather than anticodons. Meaning, if the codon is GGU, then the amino acid that the tRNA should carry is glycine. This has been determined. So, and this also already has a guide has been made in order to determine what amino acids that the tRNA will carry. And this is this table right here. RMA, if so, if the codons are UUU and UUC, then the amino acid that the tRNA carry will be phenylalanine or Phe. And if and if and if the codons are UUA, UUG, CUU, CUC, CUA, and CUG, then the tRNA that the amino acid that the tRNA will carry should be leucine or Lu. So our we have finished our activation of tRNA by forming a covalent bond by the amino acid forming a covalent bond to the unactivated tRNA. So once the amino acid forms a covalent bond to the unactivated tRNA, then that tRNA will become activated. So second step is initiation. Before that, our ribosome has two ribosomal units, the large subunit and the small subunit. And our small subunits composed of two sites, the P site or our peptidyl site and the A site or our amino acid site. What will happen in the second step is that the mRNA attaches to a ribosome so that the first codon, AUG, is at the P site or peptidyl site. So mRNA, so our mRNA will attach to the ribosome, specifically the surface of the small subunit, so that the first codon, which is always and always the AUG, is at the peptidyl site, like this one right here. Our first codon and our first colon in our mRNA is attached to the peptidyl site or P site. And then a tRNA carrying methionine attaches to the first codon. So this is our this is our tRNA. tRNA. Our first codon is AUG, right? So the anticodon that the tRNA will carry should match the codon of our first mR, the first codon in our mRNA. So if our first codon is AUG, then our anticodon should be UAC. And the amino acid that the tRNA carry should be meth or methionine. So once the tRNA attaches to our first codon AUG, then we have finished the initiation process. So let us now move to the third step, which is elongation. Elongation is the polypeptide chain grows to one amino acid at a time. So what will happen in our elongation is that another tRNA with a second amino acid binds at the A site, and then the methionine transfers from the P site to the A site. So, so in our second step, initiation, our A site was empty. Our A site was empty, right? So it is ready for a tRNA attachment. So in our elongation, our A site is ready for another tRNA attachment. So our second codon in our mRNA is GGU, and then the tRNA the I mean and then the tRNA that will attach to this codon should have the anticodon CCA and should carry the amino acid glycine or gly. So once the tRNA attaches to our codon in the A site, then the amino acid from the P site will transfer to the A site. So the amino acid methionine will transfer to the A site and will be linked to the glycine by the enzyme peptidyl transferase, forming a dipeptide in the A site. Dipeptide because it has two amino acids present or linked together. So once the methionine has been transferred to the A site, then the tRNA in our P site will become empty because the mRNA, it has already been transferred. So that empty tRNA will leave the site, and then then and then the ribosome shifts to the next codon, making its A site available for the tRNA carrying the third amino acid. So, our once the process is done, our ribosome will shift to the next codon, making our previous, making our second codon move towards the, I move in the A site, from the in the P site, leaving the A site empty or ready for another tRNA attachment. So, for example right here, our third codon in our mRNA is GCU. So, that tRNA that will attach to this codon should carry the anticodon CGA and carry the amino acid ALA or alanine. Once the tRNA attaches to the third codon, then the amino acids from the P site will transfer. Okay, let me write it down. So the amino acids in the P site will transfer to the A site and will be linked to the amino acid in the A site, which is our alanine. So methionine and glycine will be linked to the alanine by the enzyme peptidyl transferase. And once the amino acids from the P site will be moved towards the A site, it will leave the tRNA empty, and that tRNA will leave, and that tRNA will leave the site, making our ribosome shift to another codon and making our third codon move to our P site and leaving our A site empty or ready for another tRNA attachment. This process, or our elongation process, is cyclical, meaning it will go again and again and again, like it will looks like a cycle. So this will continue and continue and continue and continue until we go to the step 4, which is termination. What will happen in the step 4 is that the polypeptide chain continues to lengthen until a stop codon appears on the mRNA. So, as I have said, the polypeptide chain continues to lengthen. It will lengthen in the elongation process. The polypeptide chain will continue to lengthen, no matter how many amino acids are linked together, 200, 400, it doesn't matter. It will continue to lengthen unless a stop codon will be encountered. Our stop codons are the UAA and UAG, UAG and UGA. So once it has been once it has been appeared in our mRNA, then the elongation process will stop. So once it has been stopped, so once it has been stopped, the protein is cleaved or split from the last tRNA. For example, if our if our if our codon right here is UGA, then the process of elongation will stop. Then our code then our protein is cleaved or split from the last tRNA, which is this one. It will cleave from the last tRNA by hydrolysis reaction. So it has now been cleaved, right? So this is now our our polypeptide chain. So the the fifth step is the post-translation processing. During post-translation processing, cleavage of met or methionine usually occurs by a specialized enzyme in a hydrolysis reaction. So I think we have yet I think we have yet finished our polypeptide chain because during post-translation processing, our MET will be cleaved from the polypeptide chain through the process or through a specialized enzyme in a hydrolysis reaction. So the MET will be cleaved and then also also disulfide bridges between cysteine units can also form, like this one right here. These are our cysteine units. Or the no no no. This is our disulfide bridges between cysteine units. This is an important component of the secondary and tertiary structure of proteins because it stabilizes the overall 3D structure of the protein. And I believe that we have just finished our protein synthesis, transcription, and translation because we have now formed our polypeptide chain, which is the purpose of our protein synthesis. So, thank you for listening, and I hope that you understand my topic. And if you have any questions, do not afraid to raise your hand and approach me. Thank you.